Uh, Hi, sorry about that. Oh yes. Hi. So my name is Waldegrave, and I'm a PhD student at the University of York, supervised by Susan Martin. And I'm going to be talking about using the system developmental graph cellular automata to create network motifs. So I'll begin by kind of introducing and motivating work. So as we all know, networks occur everywhere in biology, obviously neural networks, but also gene regulatory neural networks, and then kind of these intracellular things like signal transduction networks. And then kind of protein interact, and then scaling up kind of to the macro level. We've got things like food webs and kind of social networks. And a lot of these networks have highly kind of non-random structure. So, for example, on the bottom right, we've got a picture of these kind of cortical mini columns, which seem to form kind of modules in the cortex. And certainly compared with kind of random graph generation algorithms like the Erdos-Renyi method, these are kind of highly non-random, highly structured graphs. And often that structure is the result of a kind of evolutionary and or developmental process, and there may be evolutionary pressure for that structure because the structure helps define the function of the graphs. So we want to investigate whether we can use this system developmental graph CA, which I'll come on to in a moment, in a kind of Evo-Devo method to create graphs with similar structure to biological networks. So the kind of second part of the motivation is that at last year's A-Live conference, we introduced this EGCA system, which is shown in the top right. This kind of, so you start with a very simple network, which could even be a simple node, a single node, and then it kind of grows and adds more nodes. But as you can see on the top right, the original version of the system tended to create networks which were kind of very densely connected, and there weren't actually that many different ways of wiring things up. So we've kind of introduced a new version which can specify in much more detail how connections actually made with new nodes, and that can give rise to a greater diversity of types of networks. So we want to kind of test it and see whether we can in fact produce a big diversity of networks. And yes, so I'll now just introduce the concept of kind of analyzing graphs in terms of motifs, which is really a thing from a method from systems biology. So why would you want to do this? I mean, so the kind of top-down metrics from graph theory, like degree distribution and average path length, don't give you much description of the network microstructure, and often the kind of functional properties of the system arise from that microstructure. So, for example, at the top right, there's a little kind of three-node motif, which is called a regulating mutual triad by Yuri Alon. That can implement things like kind of memory. So a stimulus to node Z is then kind of remembered by that little motif. And then in terms of kind of thinking networks as discrete, stately discrete dynamical systems, the distribution of these motifs can confer different stability on the overall network. So some might tend to encourage kind of static or oscillatory behavior, and others might tend to encourage kind of more chaotic behavior. And there seems to be evidence that evolution may push the distribution of motifs towards a distribution which encourages kind of edge of chaos type behavior. So how do we actually do this? So the kind of algorithm introduced by Milo et al. In this paper at the bottom is so say you're focusing on three-node subnetworks, triads. So there are only actually 13 of these, 13 different ways to connect on three nodes. If you ignore self-loops. If you include self-loops, there are actually 86. And that's one of the problems with this type of analysis, that there's a kind of combinatorial explosion, and so you tend to have to do it with very small motifs. So you count how many of each of these you have in your observed network, and then you create a whole bunch of randomized networks, and which importantly all have the same degree distribution, so it's a kind of fair comparison. And you calculate the z-score, the z-score of your observed network motifs versus the ensemble of randomized ones. So how many standard deviations away from the mean And then you can kind of normalize that vector of 13 z-scores to get what they call the triad significance profile, which can kind of characterize the network. So just to make that a little bit more concrete, there's a kind of very small example with a kind of unrealistically small network, but say that with your network, you would count up all of those motifs that are highlighted in yellow, 2 of these mutual in triads, and then you compare that with 500, say, shuffle networks, and then you can see on the chart that we've got the actual count and then the kind of the distribution of random counts, and then we can calculate a z-score from that. So to look at what it looks like with biological networks, here we've got 2 very different types of networks, a transcription network and a neural network, and the normalized z-score for them is shown on the right. So you can see that they kind of have a sort of characteristic triad significance profile. So the 2 transcription networks are quite different from the 2 neural networks. And they're all quite sort of far from random distributions because this is a z-score. If they were sort of random line distributions, then you expect those lines to be at 0. So now I'll just kind of reintroduce and kind of update the developmental graph cellular automata model. So conventional CA takes place on a grid. A graph cellular automata, each cell is in fact a kind of node in a graph which can be connected in any way. And that does mean that the transition rule, which updates the states, can only there's no natural ordering of the nodes, and you might also have of neighbors, and you might have a varying number of neighbors. So you can only use kind of totalistic update rules where you count the number of neighbors in each state. And in fact, you can implement that with a small neural network, so it becomes a kind of graph neural CA. So the developmental version of this is, rather than the function just outputting a new cell state, it also outputs an action, so a node can be added or removed from the graph. And as I mentioned, we use a kind of little neural network. So the input is the kind of vector of the neighborhood states, and the counts of nodes in each state. And then the output is not only a new node state, but also an action, whether the node should stay still or be removed or divide. And then the kind of additional thing we've introduced in this updated model is that there's a big kind of specification of if the node does divide, how that node should be kind of connected up. And that enables lots of kind of variation in the way it can be wired in. So just sort of briefly, this is explained in excruciating detail in the paper, but just to kind of briefly introduce it, the kind of set of allowable edges. If your original network is the blue 5 blue nodes and node C in the middle is dividing, it can have edges from its parent node C and from its parent neighbor. It can also have edges to its parent and to its parent's neighbors, and importantly, it can also have neighbors edges to other nodes which have been created in the same time step. So that means you can get whole sort of groups of nodes which all decide to divide at the same time and retain their kind of mutual connections amongst themselves. And that kind of allows these kind of 2 different routes to modular structure. So one way is that a whole group of nodes can divide and kind of keep their connections. But importantly, the decision to divide is still taken at the level of each node. And the other route is that you can have the collitive growth algorithm. You can have kind of parallel branches of a tree kind of growing in the same way, and you can get sort of larger similar modules. Again, this is explained in more detail in the paper, but we sort of implement this in quite an efficient way with tensor products. So we just work with the adjacency matrix representation, and then each update step is implemented as a tensor product of that adjacency matrix. And that's kind of important because, especially in Evo-Devo algorithms, you have this kind of double loop of the outer loop of evolution and then the inner loop of development, so it can be kind of very slow. So it's important that the developmental steps can be quite quick. So that's what a full developmental step looks like. You have your starting graph, you take the neighborhood information vector at each node, you update the graph structure, and then finally you update all the node states. So you can see in this example that therefore nodes A, C, and D all chosen to divide and keep some of those connections between them. How am I doing for time? So just to show you what that looks like in practice. This works. Yeah. So starting off with a sort of single node C graph, it goes through multiple time steps of kind of division, and then each of these divisions is kind of completely specified for a completely deterministic system, and you can see that nodes can divide in very different ways, so you can get some kind of interesting structure emerging where some nodes are more densely connected and others are more weakly connected. And you can also get things breaking off when nodes are deleted. Finally, the kind of experiments. So first of all, just the kind of proof of concept that this can create a lot of diverse profiles. The random search, so we kind of randomly initialize the neural network weights, which are the growth rules, and then we ran the system for some time steps or until it had grown to a certain size, and then we measured the prior significance profile. And so each column of this is actually a single developed graphs TSP, and they're just sorted so that you can see that there's quite a range of different motifs being the most significant one. So then we did a kind of evolutionary, target-driven evolutionary experiment, and we used the microbial GA, so very simple genetic algorithm where we basically pick two genomes from the population, we develop them, we compare their prior significance profiles with the target one, which is from a biological network, and then the loser, we kind of recombine those network weights and put it back in the population. So that kind of worked quite well. So the on the left, the target PFP is the E. coli transcription network that I showed earlier, and that is actually the evolution kind of finds that very quickly. So on the bottom, you can see the target significance profile in blue and the best evolved network on the right, in red, sorry. And then on the right, we've got the C. elegans neural network, which takes a little bit longer to find, but gets there eventually. Yeah, so kind of in conclusion, it's this kind of development of the DGCA system can create networks with a wide variety of kind of network microstructure. And in a way, this is the kind of first step, because the hypothesis is that the network microstructure is very important for network function, and that's why it's been kind of evolutionarily retained in these natural networks we see. So the kind of, and hopefully it has kind of interesting computational properties. And this DGCA system can be used to evolve and grow networks with those different properties. So yes, that's it. Thank you very much.